Hi, my name is Sabrina Zaidi, and I will be presenting signal transduction presentation on GPCR today. And the learning objective for the students after this presentation should be they should be able to understand the mechanism of GPCR and describe the structure of GPCR in details. And they should be able to explain how GPCR is activated and inactivated, and explain how GPCR affects the second messenger activity, explain the mutation and disease associated with the GPCR. As we all know that for these signal transduction, we need the cell membrane. Now let's go back to our cell membrane structure, which is composed of lipid bilayer, and these cell membrane have proteins in them which has a specific function. So membrane proteins can include from ion channels, receptor proteins, and proteins that provide the connection between the cells. We have two major classes of membrane protein: peripheral proteins and integral protein. Peripheral proteins are also known as extrinsic protein, and these are loosely attached to the membrane. As you can see here in the diagram, it shows that how loosely they are attached are numerous as compared to integral proteins. Integral proteins, on the other hand, are the main embedded proteins which are permanently attached to the cell membrane, and we have the GPCR protein, one of those integral proteins which are present in the cell surface membrane. And they can be of, as you can see here, they have the complex structure embedded in the lipid bilayer. They can have alpha helix structure, helical bundles, or beta bundle. Binding of the signal molecules to the GPCR, those membrane protein, will result in G protein activation, which in turn activates the second messengers. Now the signal communication. That's the main purpose of our all these signal transduction, that cell communicate and may be able to move to the different stages to perform the cell functions. These cell function communication involve specific and highly sensitive signal transducing processes through embedded protein, as you can see here. In one of the cells, signal molecules attaches to the receptor. That receptor conveys that message to different molecules and lead to the response. In the second cell, as you can see, the signal attaches to the receptors and activates the another molecule, which in turn can cause in two different response. And the third cell, as you can see, can have different crosstalk between different responses. And as in the fourth cell, as you can see, different receptors leads to a different response. Now we come to the transmembrane proteins. Let's just before we go into detail of GPCR, these transmembrane proteins are the protein membrane associated with different shapes and position, alpha helix, rolled up beta sheets, and they have different four types, such as plasma membrane protein receptors that act through G proteins. And we have receptors with internal enzyme activity, tyrosine kinase activity, gated ion channels, and then we have nuclear receptors that bind to the steroids and causes the gene expression. And as you can see here, we have different types of membrane proteins. These are alpha helical, beta barrel, GPCR. This is the main receptor protein we will be discussing in our presentation. Now we come to the main GPCR protein. What is GPCR? GPCR is G protein coupled receptor. These are the members of guanosine nucleotide binding protein or G protein family. This is the largest and most diverse group of member receptors in eukaryotes. Human alone have 1,000 different GPCRs, and each one has a highly specific signal to work on it. And as we can see, almost 350 for detecting hormones, growth factors, and other endogenous ligands, and almost 500 GPCRs serves in olfactory and gustatory receptors. These receptors receive messages in the form of light energy, peptides, lipids, sugar, protein, and hormone. As you can see here, this GPCR has embedded the whole cell membrane in a loop fashion attached to it. Has a G alpha, beta, gamma subunit and attached GDP. We will discuss about these now in our next slides. So the GPCR components include a plasma membrane receptor with semi-transmembrane helical structure, as you can see here, and it has the extracellular nitrogen terminal, and it has inside intracellular carboxyl terminus with different extracellular and intracellular elements. And the receptor bound G protein is the nucleotide binding pockets open up in the results, which the loss of the GDP occupying the site of the intracellular G protein, and GTP attaches, and now its activated form. Now this 7 helical structures and GTP protein get separated once it's activated, and now it goes into the different messenger to be activated, stimulate different messengers. GPCR structure, as we have just seen in the previous slides as well, that it has 7 polypeptide chain folded into globular shape embedded in the lipid bilayer. 7 segments which go through this membrane, and it has loops both inside and outside of the cell with an extracellular, we can see here, N terminal and the intracellular C terminal. And these extracellular loops have the pockets for the signals to be attached and gets activated, gets the message, and once it gets the message, it conformational change brings the conformational change in it, and the G protein binds to GTP and causes the activation of the second messengers. Now, what are the subunits of GPCR? GPCR has the units, special units, which are needed for its activation. Binds to the, and these proteins that binds to the nucleotide guanosine triphosphate GTP and guanosine diphosphate. These G proteins that associate with GPCR have 3 subunits: alpha subunits, beta subunits, and gamma subunits. The alpha subunits are, as you can see here in the diagram, that the alpha subunits is attached with the GDP molecule. When it's attached to the GDP molecule, it means that it's an inactive form. It's not doing anything. And as a sequence variation, this alpha, beta, gamma, and once it becomes activated by GTP, this GTP bound alpha subunits go separately from the beta and gamma subunits of this G protein. And it has been said that the variation in one of these beta subunits can result in individual can have hypertension problems, and the variant gene is suspected to involve in obesity, atherosclerosis. Now the G protein alpha subunit, when it binds to GTP, it becomes activated. Now this GTP G protein is called GS, or stimulatory G protein. This G protein alpha subunits binds to GDP to be inactivated. Once signal arrives, GTP replaces the GDP, and G protein subunits dissociate into 2 parts: the GDP bound alpha subunit and beta gamma dimer. As you can see here, initially it was just hanging there. Once the signal has arrived, here the signal was arriving, so it was separate. As soon as the signal arrives, the membrane changes, conformational changes happen, it attaches to this GDP, alpha, beta, and gamma subunit. Now this GDP hydrolyzes and GTP comes in, and GTP and alpha subunit of G protein goes separately from the beta and gamma subunit. They both GTP bound alpha subunit and beta gamma dimer remain attached to the plasma membrane but not to the GPCR anymore. Now they are free to interact with other membrane proteins. These G proteins remain active as long as their alpha subunits are joined with GTP. Once this GTP hydrolyzes back to GDP, it becomes inactive form. Now, what are the target for these GPCR or G proteins? The basic function for these G protein is to activate further two second messengers, which are the essential components of our functional bodily function. Very special target of activated G protein is adenyl cyclase. This catalyzes the synthesis of the second messenger cyclic AMP from the molecules of ATP, which further activates the most important enzyme, protein kinase A. This protein kinase A enzyme is very important in all our essential main essential carbohydrate metabolism, such as glycogen synthesis and glycogen metabolism. And this adenyl cyclase further causes the activation of serine and threonine residues in many mechanisms to alter the gene expression in the nucleus. As we can see here, when the epinephrine binds to the specific receptor, epinephrine again is a hormone, which is a signal here presenting, and when it is attaches to the our G protein beta adrenergic receptor, it causes the conformational change in the G protein structure. This GDP and GS alpha becomes activated, GTP gets attached to it, GTP gets removed from here. This hormone receptor complex causes the GDP bound to GS to be replaced by GTP. Now this GTP, activated GTP, again you can see, as I have said, adenyl cyclase, which causes the activation of cyclic AMP. This cyclic AMP activates the PK, and it goes further into phosphorylation of different cellular proteins which are needed in many of the metabolism, such as glucose or protein synthesis. Another target protein for GPCR is very important. One is the phospholipase C. It's a very important G protein which catalyzes the second messenger diacyl glycerol and inositol phosphate 3. These are involved, again, these catalyze the second messenger from the membrane lipid phosphatidyl inositol. This is another very essential pathway needed in human bodily processes, such as in the blood clotting factors processes. They use thrombin receptors in platelets to in the process of coagulation. As you can see here, once the signal molecules come into the GPCR, this activated causes the activation, and this alpha GTP unit become activated. This activated protein causes the further second messenger IP3 DAG to be activated, because they both need to be activated to further causes the calcium release from the endoplasmic reticulum. This calcium release is needed for the activation of protein kinase C, which is another very important element for our metabolism needed. Okay, so now we come to the GPCR families, or we can say that we can further classify this G protein coupled receptor family, especially the G alpha, into subclasses of it, which are G alpha I, as you can see, is written here. It's an inhibitory G protein, which is activated by the inhibitory signals, will inhibit the adenyl cyclase that we have in past slides. We have said that how this G protein causes the activation of adenyl cyclase, an important second messenger. Here, this G alpha I means inhibitory G protein, which causes the inhibition of adenyl cyclase. And example can be in the smooth muscles contraction. It causes the depression in neural activities. Now we come to the G alpha S. What does it mean by G alpha S? G alpha S, as you can see, the name suggests, is stimulatory. The G protein stimulatory is activated by the stimulatory hormones or any receptors, the signals, which causes the stimulation of adenyl cyclase. As we can see, this activation happening in through M2, M4 receptors, alpha 2 adrenoreceptors, and which stimulates adenyl cyclase, and they can cause the increase in heart rate, smooth muscle relaxation, and stimulation of neural activity. It is opposite to the G alpha I. Now we come to the second family of G alpha, which is known as GQ, G alpha Q, and the G protein that causes the activation of phospholipase C. We have also discussed about that important second messenger, which is involved in many of the smooth muscles contraction, and it's very important for the calcium ion influx. And we can see these activities through adrenoreceptors M1, M3, M5, which causes the smooth muscles contraction. These M1, M3, M5 are muscarinic receptors, known as muscarinic receptors. Now the fourth one, G alpha 12 or 13, which is causes the activation of Rho family GTPase. What is this Rho family? This is the rhodopsin, and which is present in the eye. It's a gene which causes the formation of this protein, and activation of this causes the rods. It is present in the rods, which is for the receiving the light perception, and it regulates the cytoskeleton and also actin. It also, yeah, Rho family activation of rhodopsin is important for the eye here. And the fifth one, which is, as we have discussed it before too, that we have G alpha, and that was attached to the GTP and causing the activation of second messengers. Though what is left was G protein, beta and gamma complex, that act as a signaling molecule by activating other second messengers on the channels, and some of those we can be seen in second messenger or the ion channels. And these one of we have seen the diseases associated with this as well, beta variation in the beta gene. And here you can see this GPCR whole molecule, which is subdivided into G alpha inhibitory, which has the specific set of responses. And now the second one, we have G alpha Q or 11, which, as we have just discussed, that it is causes the activation of phospholipase C. And the third one, as you can see, is G alpha 12, 13, which is associated with the Rho family, rhodopsin in the eye. Now, as we go further, now we have to find out what causes the G protein activity and what causes its inactivation. It is necessarily needed. Otherwise, if we just left it like that, it will causes, we will discuss those in our further slides, what can happen if we do not stop these G protein activity. And this G protein activity can be shut down by GTPase activity, which hydrolyzes the GTP to GDP and phosphate. And this is also called as GTPase switch mechanism. And this GTPase activator protein or the regulator protein signaling, are also known as RGS, are responsible for how long the GTPase switch remains open. And the catalysis of the releasing of GDP protein to allow for it to bind to GDP is done or formed by the guanosine effector, guanosine effector. Effector factor. So these are very important. These two are very exchange factors are very important. Okay, so now we have talked about GPCR. Now let's talk about how this GPCR activity can be shut down and when it needs to be activated. So there is a mechanism which is also known as GTPase switch, and this G protein activity can be shut down by GTPase activity, which hydrolyzes the GTP to GDP and PI, as you can see here. And it maintains the G protein cycle between the GDP bound and GTP bound. The protein intrinsic GTPase activity in many cases is stimulated by RGS proteins, which are the regulators of G protein signaling. So it determines how quickly bound GTP is hydrolyzed to GDP and how long it can remain active. So the intrinsic GTPase activity of most G protein is weak but is increased by regulator of G protein signaling. The signal transduction mechanism of 7 transmembrane receptors has ability to switch off. So through G alpha subunit, as it has GTPase activity that hydrolyze GTP bound to G alpha to GDP and PI. That means that it will further won't be able to join back to G alpha unit, which is causing the activation of adenine cyclase. Now this G alpha joined back to beta and gamma subunit to form heterotrimeric G protein, thereby terminating the activation of adenine cyclase. This occurs to reset as well as to prevent the continuous activation of G protein, because if we won't have this resetting or this stopping mechanism, it will continuously causes the activation of these internal mechanism, which will lead to the phosphorylation of different enzyme inside the cell, and continuous activation will lead to proliferation of abnormal cells, and it can ultimately lead to the some type of a tumor. So in this way, we have this maintenance of G protein mechanism through this GTPase switch. Now we have talked about what is the GPCR and how does it causes the second messengers to work and how it causes the function of the cell. Now, if anything goes wrong or if any mutation occurs in the process of formation of this G protein or GPCR receptor protein, it can cause abnormal disorders in the body. One of the G protein and GPCR loss of function mutation block signaling in response to the corresponding agonist. And now as you can see, one of the we can see the receptor cone opsin color blindness causes the color blindness, and it is X-linked autosomal recessive. These mutation in GPCR and G protein can cause loss of function in the normal GPCR or GTPase cycle that we have just discussed in the last slide. Loss of function mutations can be missense, nonsense, frame shift mutations that changes the normal receptor protein. One of the GPCR rhodopsin is present in the disc membrane of the rod cells of the eye, which is activated by the light, and it causes the activation of transducin G protein. If any mutation in this G protein happens, then it can result in retinitis pigmentosa. And also, any mutation in this row rhodopsin protein, we have already discussed in the back slide that it can happen through G beta subunit of G protein. And some of these mutation can lose of function of GPCR, and some are happen due to the gain of the function of the receptor. These include the failure to synthesize GPCR or G protein altogether, failure to synthesize GPCR or G protein, reach the plasma membrane, failure to GPCR or causes unbinding to the agonist, and these can causes the failure to G protein to the receptor bound signals. And again, some of more of these GPCR mutation which are happening due to the gain of function, and the diseases caused by GPCR gain of function are inherited in an autosomal dominant manner. This congenital night blindness, as I have talked about this rhodopsin, if it is absent, which is only present in the rods, not the cones, which is for the colors, this only is for receiving in the bright light. Once it's get the gain of function mutation, it means that in either GPCR or G protein, it is leading independent of agonist activation of the signaling of this G protein. It causes the congenital night blindness. There is no stopping of this GPCR, which is continuously activated and causes the activation of this rhodopsin, and ultimately it causes the night blindness. Person is unable to see in the night. Now, after doing this presentation, what I have learned that it is a very vast topic, and it still needs a lot of understanding. So I thought of this activity would help students to learn the proper way of understanding to this GPCR mechanism. So I came up with this activity that a student should create a stop motion video showing that how GPCR works to produce a cellular response, and that should be done. This activity should be done in groups, and that video that you guys gonna make, the students gonna make, it should show the following structures to clear the concept of GPCR, such as secretory cell releasing ligand or signal molecules, signal reception by G protein link receptor on target cell, and including the extracellular receptor protein, G protein, activated, inactivated, GTP, and target membrane enzyme, activated, inactivated, as I have discussed about those in my previous slides. They should mention about those second messengers, and then transduction of signal by phosphorylation cascade, including protein kinase or ATP. And there is a short video that I'm presenting here, which would help them to make something like this. Okay, so in my conclusion, I can sum up the details of this GPCR. That is, I can say now that's very essential cell surface receptors protein that respond to a variety of external signals in the form of light, energy, peptides, lipids, sugar, and protein and hormones. Ligand binding to the GPCR results in G protein activation, which is the part of this GPCR complex, and it turn stimulates the production of many second messengers. These sequence of events through GPCRs regulate many essential bodily function, from sensation to the growth to the hormone responses. Okay, I hope you like my presentation and I have explained what I was trying to explain to you. Thank you.